So I'm gonna welcome everyone to virtual breakfast this morning, and we're glad to have everyone. We're kind of winding down the season, and but we're kind of saving some of the best to last, maybe this morning. But again, welcome everyone. Let's move on to the topic for the day, and we're happy to have Dr. Erin Hill, MSU weed scientist, to talk about herbicide resistance. So I'm going to stop sharing, and good morning, Erin, and welcome.
Good morning. I'm going to go ahead and share my screen. All right, so I work in the diagnostic lab, and so herbicide resistance screening is a big part of what I do during the winter months. So now is really the time to start thinking about doing some sampling if you suspect you have herbicide resistance. But before you get into that, I just want to remind everybody that there are many reasons why weeds might not die other than resistance. And so these are some things to consider when you're looking at, do I have something that's resistant or not? As many of you know, there can be application issues. There can be issues where the weeds are not the correct size. And so the herbicides don't work as well. You can have some weeds that emerge after your application, and many environmental stresses can reduce the efficacy of weed control as well as shading. So those are some other reasons. But then, of course, resistance is one of the reasons why you might be seeing weeds survive applications that previously were dying. So what is the status of herbicide resistance in Michigan? We have a pretty detailed resistance information compared to many other states. We started confirming herbicide resistance here in 1980, and our first case was a triazine-resistant common lamb's quarters. And since that time, we've been doing more and more screening. And so what you see here on the right, there is a map of Michigan showing various shades of the counties and numbers. This is the cumulative number of species that have been found to be resistant to any herbicide within that county. And this is an interactive map that you can visit on our website, pestid.msu.edu. You can go there and click on your county and see what weeds have been confirmed to be resistant and what year we confirmed resistance. Every year after the resistance screenings are done, I write an annual report, and that gets posted on the MSU Extension News. And so we had one that just came out last week that summarizes what we've seen in the last year. The samples that we get from Michigan come from a wide variety of environments, but as you can imagine, they're concentrated in annual field cropping systems. But we do see some increasingly coming from vegetables, nurseries, and even Christmas tree farms. We are also seeing an increasing number of samples coming from out of state because there aren't any other labs that offer this service in the Midwest for sure, and I believe in the country. The bioassay samples, which is what I'm going to primarily talk about today, where we grow the weeds in the greenhouse and spray them. Our peak number of samples happened in 2016. It just happened to correspond with while I was on maternity leave, of course. But that year we had 72 samples, which is a huge number of samples to push through the greenhouse. And that was because at that point we were still questioning mare's tail or horseweed. And so we were seeing a lot of mare's tail samples coming into the greenhouse for screening, most of which ended up being resistant to glyphosate and the ALS herbicide. This past year in 2021, we piloted a molecular program where we could use tissue to test for resistance. And I'm going to talk more about that. But that brought our samples in total up to 75 for 2021. But you'll see that the bulk of those were things that we were screening to test out this molecular resistance assays. So the pilot molecular program for molecular testing is something that I worked on in collaboration with Eric Patterson, our weed geneticist. So he was the one calling the shots, and then he has a master's student, Giuliano Solfax. And this is the project that he is working on. And this work is funded by the Michigan Soybean Committee. And so what we did last year was we engaged with a couple of clients who have submitted samples through the diagnostic lab before and asked them to collect particular weeds at random when they were out scouting fields. So not necessarily weeds that we felt were resistant, but just anything to get tissue in to try out these assays. We were looking at 5 weeds, water hemp, Palmer amaranth, common ragweed, giant ragweed, and mare's tail. And we are limited to what is known when we're looking at these assays. So we are able to look at ALS resistance for all of those using these DNA extractions and assays. And we are able to look for glyphosate resistance only in the water hemp and Palmer amaranth at this time. And that's due to what's understood about the mechanisms of resistance and how we can go in and look at that in the genetics of the plant. So in 2021, when we did this pilot program, we had 61 samples submitted. So you remember I had 75 samples total. Most of them were for this pilot program. Of those, we were able to successfully screen 39, and 32 of them we were not able to screen because of a learning curve. And that's why we were piloting the program and not having it open to the public right away. We used the US mail to get samples, and that time delay with the mail we've seen lately resulted in tissue that was degraded. And so it really broke down the DNA, and we had a hard time getting the DNA out of those samples. So almost half of them, we were not able to get sufficient DNA to do the test. So now we know for sure that when we do open this up wider, that we have to have fresh tissue, and it has to come immediately to the lab for that DNA to still be viable for use. The other issue that we ran into with some of these is that the primers that are used to extract certain portions of the DNA were not necessarily specific to Michigan. And so some of our biotypes or slightly special genetics for Michigan caused some issues where we had to redesign those. But we were overcoming these same things. Giuliano and Eric have been working very hard on this. The sampling is ongoing this year to continue to work out the kinks in this. Giuliano is going out and fresh collecting the samples himself, and we're still working on streamlining this process and the protocol. So this is still something that's on the horizon and not quite available publicly yet, but we are making some headway. So the bioassays that I do in the greenhouse in the winter continue to be our most reliable source of resistance screening at this point. The nice thing about that, if we can get it to grow in the greenhouse, we can test it with any herbicide. So there's countless possibilities as long as we can overcome getting it to germinate. When we get those samples in, you may submit it and say, I'm looking for PPO resistance, or I'm looking for glyphosate resistance, but we don't only test that. We test for 5 to 7 other sites of action. And there's some examples at the side here of those types of herbicides that we include. And so we're trying to do a comprehensive screening, and then we have sometimes picked up resistance that was not expected in this way. So we can look for multiple resistance or new cases that we wouldn't detect otherwise. The only downside to this process, and why we really want the molecular testing to work, is because this is very time consuming. And so from the time I get your seeds in the fall, it can take several months before you get results. And we'll talk about that whole process, but it is time consuming, but it is comprehensive. So even as our molecular tools advance, this procedure is not going to go away, because it can help us look for new things that we don't understand at the molecular level yet. So what are our newest findings from the screens this past year? We found some new resistance in particular counties. So that county map is about to be updated. And so there are going to be some new shadings and new listings showing up. In Gratiot County, we saw ALS resistant red root pigweed for the first time. So an ALS inhibitor would be Harmony as an example. In Allegan County, we saw a multiple resistant water hemp for the first time, which is glyphosate and ALS resistant. And in Oceana County, we saw large crabgrass that is resistant to the grass herbicides, the ACC inhibiting herbicides such as Select Max. And that's only the second county where we have seen this type of resistance appear in large crabgrass. But none of these were new. We have seen them in other places in Michigan before. What was brand new to Michigan was Johnson grass resistance to the ALS inhibiting herbicides like Accent Q. We found by accident in St. Joseph County. And I say we found that by accident because we really were screening some Johnson grass samples that came from out of state. They came from the Midwest still, but they needed them screened. And so when we run these screens, we need to have a known susceptible population to make sure that our process is working properly. So I reached out to some extension educators, and we were able to get some Johnson grass seed from Michigan that I assumed would be susceptible. But when we tested it, we actually found that it was ALS resistant. Fortunately, one of the samples that came from out of state was susceptible to everything that we sprayed on it. So that was unexpected, and that's the picture that you see over here at the right is that resistance screening for Johnson grass. The other species that we had that is kind of unusual is called 6-week fescue. This is a native species that is not a common weed problem in Michigan, but there was some problems controlling it in this particular rotation in Hillsdale County. And we did find that that population was resistant to glyphosate. So those are kind of the newest findings from the bioassay screens from last year, and we will continue looking this year, and then we'll have an updated article, hopefully in the spring of 2023. So how would you go about submitting a bioassay sample? That's part of the title of this talk was that now is the time to start looking. But you really have to look at the seed to see if it's mature. You can't just assume that because it's, you know, September 15th that all the weeds you see have mature seed. That's not how it works, unfortunately. There's a lot of environmental factors involved. If you're thinking about submitting a species you've been having trouble control, we have lots of those susceptible populations to compare to for the weeds I've already mentioned, the ragweed, all of the pigweed species, mare's tail, and common lamb's quarters. If you are looking at a species that's outside of that, I just ask that you email me or call me ahead of time to make sure that we have another population that we can compare it to. So once you are looking for mature plants, usually that happens anytime between late August and October for the summer annuals. You're going to want to actually pull those plants apart a little bit and look for seeds. So on the right here, we have water hemp seed heads, and water hemp has both male and female plants. So a very common problem, one that I just got yesterday, was that someone will send me a water hemp sample and it turns out to be all male plants. Well, there are no seeds on those male plants, so we can't do a bioassay on that. So you really need to rub those seed heads between your hands or between your fingernails to look for a dark, hard seed in order to know that you're going to be able to get some results out of that. Once you have a population that does have mature seed, you want to pull at least 5 plants. More plants is always better so that we have sufficient amount of seed to work with. And you're going to package that all up. I'll show you in just a minute. We do not want any roots or soil because that can make it harder for us to clean those seeds off the plant. This is an example on the right that just popped up of a fact sheet that we have to help you successfully collect weed seeds. I've had it out for quite a while and it has helped quite a bit, but there's some photos, nice color photos in this document online that can show you exactly where you should be looking for seeds, the approximate size and color of the seeds. And you can find this on our diagnostic website, and I believe that Phil is also going to drop a link to this in the chat. For the pigweed species, I have found over time that sometimes there's confusion about what species it actually is. And so you may think that it's a water hemp or a palmer, for instance, and it turns out to be Powell amaranth, the redroot pigweed. I can tell a little bit by looking at the seeds, but it is challenging. So when you're collecting and submitting those seed heads, please leave a few leaves attached to the plant. That helps me confirm that what you think is in the bag is what is actually in the bag before we start the test, instead of finding out down the line once they're growing that this is a different pigweed. Now, we're still going to screen it, but I want to make sure that we have the appropriate species to compare it to. When it comes to packaging up your seed, you want to put those seed heads or plants in paper bags. We want to make sure that they can breathe and that we're not getting any mold developing. That makes cleaning the seeds a very nasty job, and it can compromise the viability of those seeds. You want to take that bag and double contain it. So put it in another container like a cardboard box and seal that up to either bring it to us in person or mail it in. And you need to think about that when you're sealing it all up. What we don't want to happen is that you have a potentially resistant plant that the mail carrier is then spreading seeds that are falling out of this box all the way to diagnostic services. The address is listed here, but you can find that on our website and also on the submission form. The submission form is there to collect information about where you got the sample from to help us fill that map. We will not be sharing any of your personal information or exact location with anyone beyond the lab. It's just for, we will be shading in counties, and that is all just so we can have an idea of where the resistance is in Michigan. This is a copy of what that form looks like. I actually believe the one that's up there is a little more updated right now on the website. One of the things that frequently gets forgotten is the sample location, and we're looking for the nearest crossroads, or GPS coordinates would also work for that when you send it in. If you forget to send this, I will be asking for it at some point via email or phone call. What are the fees for submitting weeds to be screened? If you are a soybean grower in Michigan and you want to test one of those 5 species I previously mentioned, then that is funded by the checkoff dollar, so there is no charge to you. Again, those species are the pigweed species, horseweed or mare's tail, common and giant ragweed, and common lamb's quarters. If you are a vegetable grower, the Michigan Vegetable Committee is sponsored screening of all species within any vegetable crop. If you have a different species or you are the different rotation type, the fee is $90 for those who live in Michigan, and the out-of-state samples are charged double. That's $180. So quickly, because I know we need to get on to the weather, what is the process that we go through? Why does it take so long to get this screening done? I told you we're growing weeds in the greenhouse to turn around and try to kill them. I find that kind of the irony of my job. I have to be very good at growing weeds to turn around and try to kill them. So to get that process started, we have to have seeds that are ready to germinate. And unlike crop seeds, weed seeds are mostly programmed to be dormant in the fall in order to survive the winter months. So we have to use little tricks sort of to break that dormancy. Sometimes it's an acid treatment or a cold treatment. Sometimes it's putting it outside and trying to expedite Mother Nature and get those ready to germinate earlier in the greenhouse. After the seeds are ready to germinate, they get planted in the growth chamber in these flats, and then we transplant them into the greenhouse, into their own individual pots. The reason we need so many seeds is that we usually need a minimum of 60 weeds that are all germinating at the same time and approximately the same size. So in order to get that, we need hundreds of seeds to work with. Once those weeds reach about 2 inches in height or diameter, which is supposed to be the ideal time for spraying, that's when we make our applications. We have a single nozzle boom sprayer that we use in the greenhouse facility. It's actually a much updated one compared to this picture, but I didn't have a picture of the new one. And then after that, we evaluate them for control 2 weeks after that application was made to determine if there's any resistance. If there are new cases of resistance, like we saw with the Johnson grass or we saw with the 6-week fescue, that requires some additional screening to ensure that we are in fact seeing resistance and the level of resistance. And so those can take a little bit longer, but we let you know that's the next steps that we're taking. So with all of that in mind, that's why it takes some 3, sometimes 4 months after you submit the sample to get results on the bioassay screening. It accounts for all of those processes that I just mentioned. And we end up giving you a sheet back, like you see in the lower right-hand corner, that will show you all of the products that were sprayed. And it will give you a rating of green, that means it's susceptible, the plant will still die, or red, it is resistant. Or sometimes we have kind of an intermediate result where the 1x field rate, the labeled field rate, did not kill it, but a higher rate, a 4x rate, which is what we also test, did kill it. And so we're starting to see resistance, but it's not a high level of resistance. So with that, I have my contact information up here, and I'll be happy to answer any questions after we hear about the weather. But now is the time to be getting those seeds into me, and the deadline for getting those in is mid-November. It's really challenging if I get them any later to get the results back to you before planting season is upon us. So thank you.
Thank you, Erin. With that, I'd like to thank everyone for attending. you can follow us on Twitter, on Spotify, the Apple Podcasts. You can follow us on Facebook. These recordings will be posted on Spotify and the podcast before the end of the day or early tomorrow if you want to re-listen to some of that information. So I'm going to stop sharing now and invite people to ask questions if they have them. I guess there's one a couple that Phil has asked, one for Dr. Hill. Are there certain areas of the state that you would like samples to be submitted from?
That's a great question. Thanks for bringing that up, Phil. It doesn't matter so much to me where, but I would say we have seen kind of underrepresentation from the Upper Peninsula. I think we've probably had less than 5 samples submitted ever from there. And so is our map really representative of the UP and also the Northern Lower Peninsula? I think, it follows well with our production systems where we see concentrations of ag. But yeah, some of those areas, if you're having trouble, we really haven't had that many samples come in the first place to have a good picture. We don't ever have, unfortunately, money to go out and just do random checks. So it's all reliant on people submitting those. And I would say the vast majority of our samples come in from agribusiness people who are out in the field scouting and doing things.
Yeah, I guess looking at the maps and look at some of the areas that I covered, that northeast Michigan is an area that, just talking with some of the farmers up there, I think that there needs to be more samples come down to more accurately represent what's really happening up there. So I would encourage the farmers agribusiness from that region to get those samples in or at least contact myself or someone to help facilitate that. I also just for logistics sake, I'm always glad, and I think all the field crop educators are always happy to help with getting those samples to campus. I mean, I know we have to rely on the mail sometimes, but the integrity of the sample is so critical, and we can help. And I'm glad to help facilitate getting those samples to campus in a timely manner, so Erin has something actually to work with. So there's another question, comment. I think it's for BJ from Phil about what does 2 to 300 degree days mean in terms of weeks, weeks in the growing season.
You're muted, BJ.
Yep. So in terms of growing degree days, so what that is, that's a measure of the average temperature given a day and how much that averages above 50 degrees for a given day. So if you're 80 degrees Fahrenheit in a day and that base is 50, that's 30 growing degree day units. So if you're looking at, 30 degree days, say temperatures average 80 degrees over a week, multiply that by 7. So that'd be 30 times 7, so 21. So 200 growing degree days, say 80 degrees Fahrenheit average temperatures would be about a week and a half ahead, for example. But that varies from day to day too. So looking at things being about a week or so ahead of schedule right now.
Okay, thank you. Just a comment, Angela put a question in about where the sign-in sheet, and Phil responded that there is no sign-in sheet, QR code for the CCA. Just get your name in correctly into the participant and send that stuff to the designated area that Phil had put in the chat box. So are there any other questions for our presenters this morning? In the meantime, I notice Dr. Manny Singh was on. Do you have any comments, anything current that folks need to know?
Okay, let's see. Dealing with the kids here this morning, Paul, so hopefully you won't hear screams in the back. So, as the weather, forecast mentioned, looks like we have a good season in front of us. We need some heat units here for crops to dry down. We have been tracking the black layer formation in corn in multiple trials on campus, and we are seeing it was little bit delayed compared to what we expected. But again, things are ahead of time, so I don't expect any issues there. I think we should be able to get to harvest maturity in time. Jeff Anderson and I have been recording a daily moisture dry down using multiple methods. So we're pretty excited about that data, and hopefully we'll be able to share that this spring. Let's see what else. So Phil put a comment in the chat box. Phil, do you mind explaining that a little bit? I wasn't really monitoring chat box. Something on soybeans.
So Manny, a couple of weeks ago, Wayne Monti, and I think Wayne is still on this morning, asked a question about whether or what would make his soybeans over the tile lines very green, whereas between the tile lines, the plants are starting to senesce and dry down. What would cause that phenomenon?
So plants are not maturing. So there's probably two different things, right? One is what we call a green stem disorder, right? So I assume this is not that, because what happens with a green stem disorder is your stem will stay green, but the pods are maturing. So it causes issues with harvesting, right? And there is multiple reasons for that. Some people thought it might be a viral thing, some sort of virus infection, but I think it's probably like a source versus sink issue. So you still typically, if you don't have enough pods on a plant, so if you had issues during the pod formation that you had pod abortion, flower abortion, if you don't have enough pods on the plant, there isn't enough energy going into the pods, so the stem end up staying green. Do you know, like, if that could be the issue going on there?
That was not the issue. The entire plant was dead green.
Okay. So that's the second bucket I could think about. That the harvest, your overall maturity is delayed, and if it's happening on top of the tile lines, I could think it might be something to do with the moisture availability. That if the plant is still sensing it has moisture available to itself, it might try to keep those leaves. So it can be like a stay green thing that I could think about, while it does not have enough pots to fill out. That might be the only thing I could think about, if that makes sense.
We didn't know it, Manny. That's why we're asking you.
Yeah. So to me, I think then if it's overall delayed maturity, it's probably something to do with the moisture availability and how far the tile lines are and all of that, right? I don't know enough about how drainage works, in terms of how far apart those lines are. But I think that's where probably it's happening, that the plant is still trying to continue while it does not have enough pots to fill out. So it's already done that. That's going on while it's still keeping more energy that it doesn't have anywhere to take to.
So Leon has a comment. He says plants over the tile lines have their roots drawn down deeper and tend to produce more and longer in dry years.
Yeah.
We didn't know if it was just a matter of more opportunities for air and better growth over the tile lines, but it's one of those things where we were hoping for some clarification.
Yeah, so that's what I'm thinking, that it's probably that better growth, but it doesn't, because of the relatively dryness, maybe early on, it doesn't have enough pots where the energy can go on. So it's still staying green because it doesn't have anywhere else to send those sugars. That's what I would think, but I can try to look a little bit more into that and see if anyone else has noticed that. But I would associate again this with the dryness that we had in August. Probably if this field did observe that, and that would explain what's going on.
Okay, thanks, Manny. Any prediction or any thoughts on what % of the crop in Michigan is black layered?
I haven't seen the recent USDA report here. I would think it's probably hanging in between like around 20 to 30%. It's going to depend on the planting date and relative maturity, right? And that's what we are tracking right now, that the early planting and a relatively short maturity is getting close to black layer. It was behind where we expected. We typically use there's a free online tool called U2U, useful to usable. I would recommend everyone to go and check that tool. You simply enter your location, planting time, and the hybrid maturity, and it spits out the black layer estimate for you. So that's a good tool to use while evaluating where the field might be.
Okay, thanks, Manny. Another question in the chat for Erin. She answered it in the chat, but for the folks online about in the state, which weeds are now officially resistant to glyphosate? Yeah,
so this is unfortunately an increasing list, but by far and wide, the most widespread resistance to glyphosate we're seeing in mare's tail or horseweed. We also see resistance in palmer amaranth, water hemp, common ragweed, giant ragweed. Last year, we found a population of annual ryegrass that was resistant. And then this year we added 6 weeks fescue to the glyphosate resistant list for Michigan. So unfortunately, that list is getting a little longer. I would say those two grasses and the ragweeds is not widespread resistance at this point, but mare's tail for sure is widespread because of the wind dispersed seeds.
Thanks, Erin. Clay has a comment: with warmer temperatures and drier conditions forecasted, should we delay wheat planting? And I think that's a question thrown out to everyone. My answer would be, don't delay, get it in the ground. There's no saying mud the oats and dust the wheat. Manny?
Yeah, so we have done a lot of work recently on planting time. What is the delay and what is the penalty with a delay in planting? I think the article came out on MSUE News last week, so please check that out. But what we are seeing is that the decline or the penalty starts soon after October 1st. So do not delay it. There is no benefit to like sort of jumping the gun, planting too early. And I think we are past that window. So if you can, I would plant now. One thing we're finding out is that there is no benefit of really pushing to high seeding rate, especially under early planting. We might benefit. It might help with the lodging issues and things like that later on. So if you are like up in 1.6 million, try to cut it down 20-30%, maybe throw a strip in there and see what happens. That potentially can benefit along with early planting.
Okay, thanks, Manny. There was a comment about the tile lines, extra iron being in drier site from the tile. And then there was one about black layer. What is black layer? And I think Phil answered them very well in the chat. Phil, do you want to comment on that?
Well, let me start with the black layer for corn. That is a physiological maturity of the corn plant. And so those kernels that are on the cob will have a thin black layer at the end of the kernel. And if you pull one of those off of the cob and kind of snap the end, you'll see a small black layer at the end of that kernel, which means that there are no more nutrients being transferred into those kernels by the plant. And so it's mature, and the corn that's there is what you're going to get. So, and for the iron, that's a hard one for me to answer, because I'm not sure that iron is the critical nutrient over the tile lines that would make it stay greener longer. Manny, I would defer to you on that, but I would say that without having tissue tests for those plants, it's going to be difficult to tell if there are or are not more of those micronutrients like iron available for the plants.
Yeah, I don't have a good answer to that either. I mean, it could potentially play a role, and nutrient might be part of it. I believe it's driven by, again, moisture, how much moisture you have available within that profile, right? And again, a lot of nutrients do diffuse with water, right? So if you have availability or access to more water, nutrients can be like a confounding factor on top of moisture. But yeah, I'm not sure if iron is a big player there. And going back to the black layer again, I think Phil explained that clearly, very nicely. That's when your yield is formed. Again, if we are running late, a frost were to occur, if the corn is already at black layer, you are not going to lose any yield because your yield is set at that point. The only physical thing that happens after that is the dry down, right? So around black layer, we are dealing with about 30% moisture, also 30 to 35. And we want to bring it down close to 20 or below for harvesting, right? With the energy costs where they are, we want the field dry down. So that's the only thing that happens after black layer.
Okay, thank you. There's no other questions in the chat box, and I think Dr. Chilvers has left us looking at the participant list. So are there any other comments that anybody would like to add this morning? If not, again, I'd like to thank everyone for joining us this morning. Thanking Dr. Hill and BJ for being with us this morning. Please join us next week for our last session with Dr. Christy Sprague. So with that, have a good day, everyone.